And introduce Professor Tatiana Novikova to speak with us about Mueller Polarimetry for tissue diagnosis. This is an area she's worked in for decades, and personally an area that she taught me about during my sabbatical when we worked together as a D'Alembert Fellow. She's currently leading a group of applied optics and polarimetry at the Laboratory of Physics of Interfaces and Thin Films of CNRS at École Polytechnique. This is in Palaiseau, just outside of France, just outside of Paris. She also has a courtesy professor appointment in the Department of Biomedical Engineering at Florida International University. She's obtained degrees in applied mathematics from Moscow State University, physics and mathematical modeling, also in Russia at Moscow State University, and physics from University Paris-Sud in France. So the research interests are in Mueller polarimetry and polarimetric instrumentation. Even in the short visit to my lab in doing eye polarimetry, she's given us critical pieces of advice just during the lab tour. A true expertise. She's a member of the editorial board of the Journal of Biomedical Optics. She's right now co-editor of a book on polarized light and biomedical imaging and sensing. This is one that came out in 23. I know you'll have a new book project as well that you were telling me about at lunch. She's a fellow of SPIE in Optica and the recipient in 2020 of the very prestigious SPIE Gigi Stokes Award. Thank you, Professor Mansurapur, for being here. He likes to call all of us working in polarization imaging very polarizing people. I consider that a compliment, and that's good company to be in. On a personal note, I just want to say that in the career, since we have many students here, in the career you will have several pivotal people that when you look in the rearview mirror, you will say they made a difference, and so on and so forth. And when I had my first daughter, which is a big identity shift. The second and third, right? Easy by comparison, a big identity shift. And I was coming back to work. I attended a small workshop in the Netherlands, and I decided I really want an international collaborator. I don't have one. And I just forced myself to introduce myself to people. Hello, I'm Meredith Kopinski. I'd like an international collaborator. And Professor Novikova is the one that said, tell me more, what do you know? What could we possibly work on together? So thank you so much for that. It's made a difference to be your colleague. Thank you for being with us today. And please join me in welcoming Professor Novikova.
Thank you very much for the introduction and for many nice words you told to me. And today I want to.
I'm so sorry to interrupt. Turn the switch on your. I think you turned off the mic. Oh, I think it was on in your pocket. We need to make sure it has the green light for the mic. Yeah, I saw the green light.
Okay, I think maybe because it's pointed down. The other. I'm
so sorry.
Oh.
Thank you so much. No, amazing talk.
Actually, it's my second time at this university at exactly this colloquium. It was 10 years ago, also by invitation from Meredith. And this, let's say, I will present you our recent advances, what we did in this 10 years. And yeah, like title said, we are doing imaging Mueller polarimetry for biomedical applications, and I will show you two applications, micro and macro scale. This is the outline of my talk. Just briefly recall you the basics of Stokes-Mueller formalism. I think the people here know it very well, so I will not spend too much time. And a few words about the imaging Mueller polarimetry, and then I will present you these two case studies, our ex vivo preclinical studies of imaging with polarized light. The first problem is the problem of a preterm birth risk, and the second, we have now a big running project on brain cancer detection, and then conclusion and perspectives. So, as you know, Stokes vector, it's a vector which can characterize the polarization of our light beam, and it's composed, you can write it differently, but the beauty of this representation that it's four real numbers which represent measurable quantities, intensity. The first component is the total intensity, and second, third, and fourth, it's the differential intensity in a linearly polarized horizontal vertical plus minus 45 and right and left circularly polarized light. And normalizing by first component and taking the last three numbers as a Cartesian coordinates of a point, we can plot this Stokes vector, which should represent, let's say, it's not any arbitrary four numbers. They should obey this constraint, which you see at the bottom, degree of polarization. We can plot this in Cartesian coordinates on a surface of sphere, which is called the Poincaré sphere. And completely polarized light represented by the point on the surface. And if light is depolarized, we go deep inside, and completely unpolarized light, it's a 0, 0, 0 point. And we know how to describe our probing light beam in terms of its polarization property by Stokes vector, which we can measure. And in linear optical system interacts with this probing light beam and modifies the state of polarization, even partially can scramble it. So we can represent it by this simple equation in terms of Mueller matrix algebra. So the modification of input Stokes vector by sample into the output Stokes vector. And actually this 4x4 real valued matrix, it's a matrix which we measure, which contains all polarimetric properties of our sample. But we have to be careful, because it's not only sample, it's our instrument plus sample. Because if we change the configuration, angle of incidence, wavelengths, transmission versus reflection, the coefficient of Mueller matrix for the same sample will be different. In general principle of any polarization technique, so we have a source, then we assemble optical elements which polarized our light, usually polarizers, wave plates. Then here schematically, like elliptical polarization state interacts with the light beam interacts with the sample. Polarization is modified, then we filter it, we project it on assembly of optical elements, which we call polarization state analyzer, and we detect the intensity. And usually, in order to get the polarimetric properties, we need to measure more than one polarization state projected on a sample. And an example of very simple polarized light imaging technique, it's just polarized microscopy. Our PSG is a polarizer, and PSA also polarizer, but they are crossed. And when we put sample between them, we can get a very colorful images. Like you see the section of deceased ivory with the black points which represent the eigenstates, yeah, because it means that the sample doesn't modify. If we send a vertically polarized, linearly polarized light, it stays, sample doesn't change it. And it's completely blocked by the analyzer. So we have these dark points. And another example, it's the imaging where this crossed polarizers, liquid crystals. But it's, let's say, difficult to quantify, to extract some quantitative metrics. And to measure all elements of Mueller matrix, you need to build an instrument which is called the Mueller Polarimeter, complete Mueller Polarimeter, which measure all 16 elements. And obviously, to measure 16 elements, you need to do at least 16 measurements. And the idea is simple. So you shine different polarization states. They interact with the sample, and then you project on usually the same polarization states by varying them. Yeah, the set of 4 input Stokes vector and set of 4 Stokes vector for the polarization state analyzer. And what you measure at the end, you measure the raw intensity data matrix B, which we represent as a product of this matrix W. Columns represent the Stokes vector of your probing light beam, and A represents the lines, the transpose Stokes vector of this basis for the measurements. And final results, you just need to invert matrices A and W. And when you design your instrument, obviously you should look to get a well-conditioned matrices, because if the determinant is very small, so inverting, you will get a propagation of noise and your measurements will not be good. We implement in our complete Mueller polarimeters the eigenvalue calibration method, which was invented by our former PhD student Eric Compa almost 27 years ago. And the advantages of this method that it doesn't require the direct modeling of the instrument and any specific samples. And just to show you the examples of 2 polarimeters which we use in the studies, case studies, which I will present. The first one is a multispectral wide field imaging Mueller polarimetric system. It's based on the ferroelectric liquid crystals. So we have a halogen lamp, our PHG composed of polarizer and 2 liquid crystals. And the PSA, it's the same assembly of instrument, but in a reverse order. So these electrical ferroelectric liquid crystals, they are electrically driven, so the switching time is quite fast. And we can do, the measurements are sequential, so we send one polarization state and project on 4 different states. Then we change the input polarization state and again project on 4 polarization states. So the basic version of this instrument, it's about 8 seconds to do the measurements. Because of liquid crystals, we are limited by the visible wavelength range because they do not perform well in UV and far infrared. And it's a wide field imaging, so we do not do scanning, so we just take this polarimetric photo of a sample. And another instrument, it's a Mueller microscope, which operates in transmission configuration. It's the same idea. So we have a PSG and PSA based on liquid crystals, but we have a microscope objective. We can use 5x and 20x, which produce a microscopic images, Mueller matrix images of sample under study. And let's say, okay, we measured 16 elements of Mueller matrix, and how to interpret them? What do we do with them? Standard physical approach, you have experimental data, experimental Mueller matrix, so you need to build electromagnetic model which relates the physical property of your structure to the measured elements of Mueller matrix. But for biological tissue, which we are studying, to build an exact electromagnetic model, it's a super difficult task. Yeah? So we adopt the phenomenological approach. It means that we just use matrix algebra. We decompose the experimental Mueller matrices into the assembly of simple Mueller matrices of basic optical elements, which have a clear physical interpretation. And in biophotonic biomedical community, the most widely used decomposition is a decomposition developed by Liu and Chipman, which says that any physically realizable Mueller matrix can be represented as a product of the matrix of diattenuator, retarder, and depolarizer. And just to show you an example, so with our imaging wide field imaging system, we imaged an ad hoc sample, which is composed of mica plate, container with milk, and tilted glass. So we assume that tilted glass should produce diattenuation. quartz plate should be retardant, and milk, highly scattering, should depolarize. What we measure, you see very colorful images of Mueller matrix, but it's difficult to say what is what. And when we applied pixel-wise Liu-Chipman decomposition, this was measurements in reflection, and you see that the results are meaningful. What we expect to be depolarized in milk, yeah, we see on the first image of depolarization, white square of this container. Retardance is present at the quartz plate location, and dichroism tilted glass. So it means that we can use this polar decomposition, which was initially suggested for the measurements and transmission, but from practical point of view, we can apply it to the measurements and reflection. And so the first problem I will present, it's assessment of the risk of preterm birth. It's a work which was done in collaboration with Professor Jessica Ramello-Roman from Florida International University. What is preterm birth, spontaneous preterm birth? When baby are delivered before 37 weeks of gestation, it's called a spontaneous preterm birth. And actually, worldwide, you see the map, it's between 11 and 50% of babies are born prematurely. And this preterm birth is the highest cause of infant death. Etiology is unknown, the exact. Sometimes it's infection, inflammation, genetic predisposition. But biologically, what does it mean? It means that uterine cervix, extracellular collagen matrix of uterine cervix, lost its mechanical property. And if we look at this particular part of uterine cervix, the collagen is highly arranged and it's circumferential to hold the weight of the developing fetus. And pregnancy progresses, the cervix weakened. You see, this is the plot for the mice measured cervical stiffness. So before delivery, it starts to become softer. Then after delivery, it repairs and recovers its stiffness. The arrangement of, as I told, collagen is circumferential around the cervical os. And the same data from human cervix, so this is the MRI image, and you see that indeed the collagen is arranged circumferentially to keep this mechanical load. But for a moment, let's say there is no reliable clinical tool to measure, to assess the risk of this preterm birth. We know that state of collagen affect this risk. If collagen is softened, that it means something goes not in a correct right direction. So are we able to measure these changes in collagen? Collagen, it's optical anisotropy, yeah, and it's known that polarized light is very sensitive to optical anisotropy. So the idea is to use this Mueller imaging polarimetry for assessment of anisotropy of collagen in the uterine cervix. And so this is the cartoon which shows, let's say, how uterine cervix changes during the pregnancy course. And you see that from distal to proximal end, progressively, let's say, the collagen chain breaks and cervix soften. And the main question, because let's say before building this instrument, we need to understand, are we able to detect these changes early enough before delivery? Because the technique is quite, let's say, shallow penetration depth. And to address these questions, we did our studies in mice model of pregnancy. So you see the cervix of mice, of pregnant mice, was sliced after the animal was sacrificed. It was sliced in thin slices about 70 micrometers nominal thickness, placed on the, from top to the bottom, placed on the microscopy slide. And we imaged these slides with the Mueller microscope, which I showed you. And the measurements, they were done at the beginning of pregnancy, day 6, and 1 day before delivery, 18 day, because the whole cycle takes 19 days in mice. And you see the microstructure of uterine cervix of mice. On the left, on the right, you see the X-shaped os, because in mice it has this specific shape, and vaginal fornices. Then you see the intensity image as we detected with our microscope. And then we apply this decomposition and Leu-Chipman decomposition, and you see the image of scalar retardance. And we see the red circle around the X-shaped os, which confirmed by the SAG microscopy image. You see the green, which is the gold standard for collagen imaging, that indeed this optical anisotropy comes from the collagen in this section of mouse cervix. And in this table, you see the columns, they correspond to the early pregnancy, day 6 and day 18, and the lines, they represent just intensity images, linear retardance, depolarization, and azimuth. And we see that early pregnancy from top to the bottom, it's a different upper part of uterine cervix and lower part. Collagen is arranged circumferentially. And 1 day before delivery, its structure completely lost. Yeah, we don't see any retardance. It drops up to 0. And the azimuth orientation of optical axis is not circumferential, but completely randomized. When we so the idea is that at 1 day before delivery, the extracellular matrix of collagen is modified throughout the whole cervix. So, and we can detect it in the scalar retardance, linear retardance images. So it gives us a hope that if we will develop such instrument, and Jessica, she's working on the development of insertable Mueller colposcope. Yeah, this instrument is already in preclinical studies that analyzing these images, the gynecologist can predict if pregnant woman is at risk of preterm birth. And statistical analysis of this scalar retardance map shows that the distribution, you see the red, it's for day 6, and blue for the late pregnancy. And what we did a 2 Gaussian fit of these distributions. And you see at the bottom, like blue columns, they show the position and width at different days of pregnancy of the first Gaussian, and there is not so much changes. But the red columns, they show the position and width at different days of pregnancy of second Gaussian. And we see that it drops the position shifts and the width changes. So in conclusion, we can say that this is very preliminary studies, but we showed that polarization is sensitive to modification of collagen arrangement in the uterine cervix during pregnancy. And we can use this polarimetric signature as a quantitative marker for predictive marker of preterm birth risk. And the suggested metrics for this 2 Gaussian fit doesn't require any manual mask selection region of interest. We just analyze the whole image and we can get the idea about this. So, and the second project which I want to present you, it's a Horao project which we carry together with the University Hospital and University of Bern in Switzerland. It's visualization of brain tumor border and fiber tracts of tumorless tissue during neurosurgery. And again, brain cancer statistics, how common is this cancer? You see that it's quite rare, 1.3%. But it's scary because, let's say, our brain is our processor and everybody wants to keep it going on. So that's why we would like to find a good treatment for this disease. And if we look at the statistics, let's say, as a function of gender and age, we see that in young people, the incidence rate is very low, but with aging population, the incident rate grows and with slight prevalence in a male. And what the most deadliest brain cancers like glioma and glioblastoma, they grow from the native cells of white matter, astrocytes, and let's say they are diffusive and quite aggressive. And the first line of treatment is surgery, and survival rate may double if the surgeon remove as much as possible. But contrary to the cancer in other parts of our body, where surgeons have safety margin, that they can cut a bit more tissue. In brain, they do not have this possibility because they should preserve the tumorless fibers, because if they make surgery and person becomes blind or speechless, so it's they should avoid this situation. So for them, it's very critical to see where is the border of tumor during the open brain surgery. But despite the fact that you see on the right panel that preoperative MRI image of high-grade glioma clearly shows where the tumor is located, when they open the brain, big question mark, like where to go. And current solutions which exist to make an intraoperative MRI, but this is, it takes long time, it's expensive, and this is a large footprint equipment, and not all hospitals have these facilities. Another solution is optically based. It's called 5-ALA assisted brain tumor surgery. The patient takes pill several hours before surgery, and these chemicals accumulate in the tumor zone, and because of tumor metabolism, they produce fluorescent molecules. And during surgery, and this is already industrially implemented option, surgeons shine the light which excites this molecule and put the filter on the detected window of detector, and you see this pink spot. So this is fluorescence from the tumor cells. But the problem is it's kind of gold standard for glioma, but it doesn't work for glioblastoma. Not all types of tumor, and it works for well-differentiated tumor, because if a tumor cell concentration drops gradually, so you just your detector doesn't have enough sensitivity to detect the intensity, but tumor cells are still there. So they need another solution, and we think that our collaboration starts that we suggest to use the polarized light imaging. And these are our first proof of concept studies. It's a formalin fixed, thick human brain specimen from the autopsy of anonymous donor from the pathology department of Bern University Hospital. Thick, it means it's about 1 centimeter. And you see two adjacent slides which were, you see the red dotted line, it's a coronal section. brain hemisphere. And these sections, one went for histology analysis and another for polarimetry imaging. You see this whole mount section, thin section stained with a special protocol, silver impregnation, which is very good for visualizing this myelinated nerve in the white matter of brain. And these are the images which we get with our wide field imaging system. Intensity in grayscale, depolarization, scalar linear retardance, and azimuth of the optical axis. The measurements were done at 550 nanometers. And when surgeons first saw this image, they told us, wow, such a contrast. You see that white matter indeed is very depolarized, and we see clearly the border between gray and white matter. It's birefringent white matter because we see value of retardance which go up to 20 degrees, while gray matter is absolutely not birefringent. And we see the azimuth of optical axis is very well correlated with the direction of nerve fibers. I will show you on the next slide. When we took this image map of the azimuth of optical axis and we zoom in particular zone, which is called U fiber, which connects the neighboring zone of the cortex. So what did we plot? At each pixel of our image, we plot the unit vector with the orientation of angle of azimuth of optical axis. And the specific regions, A1, A2, A3, A4, and A5, it's within the gray matter. You see the random orientation of optical axis. And we selected the corresponding positions at the zoomed image from the microscopy of this thin histological section. And the correlation is very good, let's say, where we expect to have a vertical orientation, plus minus 45, horizontal, or completely random in this zone A5 and B5, because let's say a special resolution of our instrument, about 100 micrometers, we cannot resolve fibers, single fibers within the gray matter of brain. And just to show you another example, we took in the red square, you see the specific part of brain, which is called corpus callosum. Actually, it's a bridge which connects two hemispheres of our brain. And why we decided to image this specific part, because it's very well known that the fibers, they follow this arc. Yeah? They go to connect two hemispheres. And we imaged this sample by rotating it 10 degrees, and we you see this is the orientation of optical axis. So it goes slowly from 45, then it becomes green, and then red vertical orientation. And so we are quite confident that with this technique we can visualize fibers in brain. And this is the example, another example of the whole brain section, formalin fixed, and this is a tumorless specimen. And again, you see the image of intensity, the image of depolarization, retardance, and orientation of optical axis. But of course, we should keep in mind that these are flat, formalin fixed, so it's just first step to be sure that we can see something in reflection. And we developed also, because it's fine when we see some contrast, but if we say that this is tumor, we need a ground truth, yeah, histological confirmation that the contrast which we observe really corresponds to pathology state of the tissue. And so we developed, you see on the left and on the right, the protocol for histological validation of our images. For each image of polarization, we have the corresponding histology image. The next step, we did experiments on a fresh calf brain So we didn't kill any calf. In France, it's food. We just went to local butchery and we bought what they call cervelle de veau. To exclude the impact of formalin, so we cut it and we imaged another zone which is called cerebellum, and you see this dendritic type structure. And again, we observe very good contrast in image of depolarization. And when we applied a binary mask, we put a threshold on depolarization 0.87 and put it on the azimuth just to exclude the random orientation of azimuth in the gray matter. From A to B, we really see this U fiber orientation, horizontal minus 45, vertical plus 45, and again horizontal. Next experiment we did again with this fresh calf brain specimen. We explored the impact because, okay, we took this, no blood, it's just a piece of food from the butchery. But in real conditions, there is always blood in surgical cavity. So what we modeled this, we just opened corpus callosum, two hemispheres of this calf brain, and we created an artificial pool. We inclined it a little bit, put a piece of tissue and filled it with the blood. And you see we did the measurements at 550 top row, and then we change the wavelength 650, it's the bottom row. And on the right, you see the absorption spectra of hemoglobin. It has a peak of absorption at 550, and indeed our azimuth is destroyed. We do not see the orientation in the zone of this blood pool. But it becomes completely transparent in the intensity images, and we recover the orientation, vertical orientation in the image plane of the fiber brains and corpus callosum. And just to show you the examples, this is preoperative MRI image of glioblastoma of grade 4, excised specimen, which was just taken by a surgeon to imaging stage of our instrument. This is the photo, and on the left you see the azimuth optical axis, and it's not completely randomized, but it's kind of mosaic. Yeah? So we have some small domains of orientation, but there are no fibers going through. And this is 3 more examples. Top row, it's the tumorless formalin fixed human brain, and middle and bottom row, it's 2 different types of tumor, glioblastoma and oligodendroglioma. And how to read these images? You see on the left, it's a histological truth. That's what medical doctor told us. Green, that the top layer is completely tumorless sample. Then middle red, it means tumor center. The concentration is more than 70%. And infiltration zone, when the concentration of tumor cell is less than 70%. And we see that the depolarization is strong for all samples, but azimuth is completely erased. And retardance in the middle row drops. At the bottom, it's still present, so it's something to explore. But at least it proves that these polarimetric maps, they contain information on presence or absence of fibers. And so we developed, up to now, it's already in place, the pipeline ex vivo, what we did, a lot of studies. From autopsy or neurosurgery, we get the samples, we image them, we do data processing to obtain the image of azimuth depolarization and linear retardance. Then neuropathology annotates, creates a mask, healthy tissue, tumorless, infiltration zone, and we create the data set for machine learning algorithm. And there is a machine learning model trained on this annotated data, which makes a diagnostic segmentation of scene which we observe. And you see the examples of 4 images, like first 2 columns, it's a green color, it's a healthy tissue, and red, it's a tumor. A statistical analysis shows that you see this red histograms, it's for tumor, and green, it's for healthy. So we observe that mean value of depolarization slightly drops in the tumor zone. Retardance also drops for tumor, and azimuth variability becomes very high in the tumor zone. So indeed, all these 3 polarimetric markers, they're important for delineating tumor zone. But then we need to go to operating room. So we developed a compact version of this lab prototype. You see the photo, and we attach it to mechanical arm. The principle is the same, so we didn't change, we just make it miniaturize this instrument. So we measure intensity, we calculate Mueller matrices, and we calculate the maps of polarimetric markers. And so we did first in vivo measurements in the surgery. So you see this instrument is attached and covered with the sterile plastic cover. And these are the images which we obtained. Intensity, azimuth, depolarization, and retardance. And this is just gray matter. And these first trials, they showed us that speed of acquisition and processing is critical, because even if the patient is fixed, anesthetized, but there is a heartbeat and brain pulsates, so we need to go fast if we don't want to have a blurring images. So with all this, we updated the instrument design. What we have now, we replaced our PSA block by 2 polarization sensitive camera with the beam splitter, and first camera filters linear polarization states, and second we put a quarter wave plate in front of it. It's for filtering of circular polarization states. And with this instrument we are able to get up to 4 frames per second. We use the red light. I showed you the reason why we do this. Field of view can change from, we have an autofocusing system from 1 to 10 centimeters, and we have all these features implemented. So this is the example of in vivo images of brain surgical cavity acquired with this new updated instrument. We see the color photo, intensity grayscale, and the mask yellow, it's region of interest, it's the bottom of cavity, and these gray patches, it's the regions of gray matter. And when we applied this mask on the images of depolarization, retardance, and azimuth, and feed it into our machine learning algorithm, so the diagnostic segmentation showed that, let's say, all bottom of this cavity, it's a tumor, brain tumor tissue. Another example, we imaged the spinal cord tissue during spine surgery. And you see by blue arrows, we show the midline of the spine. And on the side, there are segmental spinal nerves, which are inclined at the angle of about 60 degrees. And what images, I just applied the mask for this zone of interest. And you see that it's depolarizing. There is retardance, and the orientation of these segmental spinal nerves, it's exactly what surgeon expects. So they were very happy with these images. And we developed also the graphic user interface. It's still, let's say, in process. We are making measurements to complete our database and to make our segmentation even more precise. But it means that the surgeon will see this And when I say that 4 frames per second, it's not only acquisition, it's acquisition plus data processing. So surgeon will see this at the bottom, polarimetric maps, but they say not too much to him. The MRI preoperative images. And then you see on this grayscale, if he puts the pointer, the algorithm reconstruct fibers, if any, from the azimuth of optical axis. And on top, let's say they want to have, if there are no fibers, the heat map of probability that the tissue is a tumor. Like the more orange the color, the higher probability that this zone of tissue is a tumor. And what's next? What are the perspectives? We know that in surgical conditions, we do measurements at normal incidence. We cannot move the instrument to play with the angle of incidence. And at normal incidence, we do not observe any diattenuation. So tissue is depolarizing and birefringent. And we showed that, let's say, if we make 3 assumptions that no polarizance, no diattenuation, and no circular diattenuation and circular retardance, the Liu-Chipman decomposition analytical form can be simplified as a product of these 2 matrices, matrix of linear retarder and depolarizer. And we show that, let's say, there is a simple analytical expression which allows us to calculate retardance, orientation of optical axis, and coefficient of linear depolarization. And when we implemented, of course, let's say we took the full Mueller matrix, but we just dropped last row, and we use only 3 top row to simulate this partial Mueller polarimeter. And we see that top row on these images, it's a classical Liu-Chipman decomposition of 4 by 4 Mueller matrix. And the middle row shows the retardance and angle of orientation and depolarization, which is calculated by using the simplified Liu-Chipman decomposition of a rectangular 3 by 4 Mueller matrix. And at the bottom, it's the orientation angle, which we can calculate just doing Stokes polarimetry, sending the circularly polarized light and calculating, well known to all of you, this angle of polarization and degree of polarization. And the visual takeaway message, we just got this video, so I am happy to show you. So brain has structure, healthy brain. It's when tumor grows, it grows chaotically and breaks this other structure of healthy brain. And neurosurgeon, when he or she is doing surgery, relies only on his skills, like where to remove. They have a special tool, ultrasonic aspirator, and they do not see fibers. So our idea, our solution to implement into this surgical microscope, the polarimetric imaging facility, polarization state generator and polarization state analyzer, which will send polarized light on brain. It will be diffused, scattered, and went through the detector, through polarization state analyzer, and then go to data processing unit, where we do data post-processing, the decomposition of Mueller matrix, the segmentation of obtained image using these polarimetric maps. And at the end, the surgeon will have the AI generated heat maps. He will see where to put his aspirator CUSA tool to remove the tumor tissue, because they are removing in a piecemeal fashion. And to conclude, I showed you that imaging Mueller polarimetry is sensitive to tissue microstructure. It can be microscopy or wide field modality. We are operating in a visible wavelength range, so it's harmless to patient. can be implemented at relatively low cost, and it holds strong promise for in vivo applications. And with this, I would like to acknowledge the funding, the contribution of my colleagues from LPCM, Florida International University, and the whole team of Harao project. And thank you for your attention, and I'm ready to take your questions.
Thank you so much for the talk. Questions, please.
Thank you very much. I'm just curious, so in terms of the applications, the areas in the human body that you're using the polarimetry to diagnose medical conditions in, is it more driven by your motivation around what type of conditions you're interested in helping to advance the medical research on, or where this type of polarimetry would be most likely to be most successful?
I would say that the first condition, if we do not talk about microscopy, with the instruments at hand, we should have direct access, yeah, because we do not work with the fibers, so we cannot image an inner cavity. Travis showed at Photonics West very nice presentation, yeah, the first steps towards endoscopic polarimetric imaging, so I'm looking forward to see your results. So for a moment, what we have, we should have direct access, either uterine cervix or open brain surgery or spinal surgery. People try to, so it's the first necessary conditions, yeah. And the story actually of our collaboration with the Bern University Hospital, very interesting. I already told to some students during the lunchtime. It's not we found them, but it's rather they use a very, for me, unconventional way. They put their challenge on a specific site, which is called xerox.com. And they explain very nicely everything, like no visual contrast, but there is structural contrast, and everybody who knows technique, which is able to visualize the structural difference, you are welcome to apply to this competition. Before they collect money, 50,000 euro by crowdfunding, yeah. And our former PhD student, he applied to compete in this competition, and when he went to the semi-final, he contacted us. He explained like there is such an interesting competition, and I would like to suggest this technique. We helped him to prepare the final presentation, and he won. He got his 30,000 euro, and we got this contact with the medical doctors. And my personal experience that it's very important, maybe also to answer partially to your questions, what is the driving force? You need to have someone whose skin is in the game as your partner. Because if we come to the hospital and say to the doctors, like, we have this technology, would you like to try? Most probable answer will be no. We are, let's say, we have a lot of our problems, we don't need anything else. And when your partners are interested, when they, let's say, push together with you the technology, then the chance to get the results is much higher than when it's only you.
Hi, great talk. I had a question about your frame rate. So early on, you mentioned, the brain, the pulse, the heartbeat, and all that. You were measuring. You had 16 images in 8 seconds, right? And later on, you mentioned how the Lou-Chipman decomposition can be simplified. Making your image processing probably a little bit faster. I was wondering, like, what frame rates do you achieve, and what is the kind of the minimum viable frame rate for this in vivo imaging?
Thank you for the question. Maybe I was not very clear, but with this updated design, when we understand that we should go faster and we use two polarization sensitive camera, we don't need to make 16 measurements. We are doing only 4 because we just modulate incident polarization like horizontal, vertical, 45, circular, and the filtering appears simultaneously. And so these perspectives which I showed, like we can extract this information even without measuring the complete Mueller matrix. It's for future when we can get rid of camera with the quarter wave plate. Let's say we will just shine linear polarization and that's it. So this is the no, I'm sorry. For this, we will still shine 4 incident, but the instrument will be more compact. And now the work is in progress. We are showing that it's not yet published, but we applied for patent. We are quite confident that we can extract this information even only sending linear polarization states. So we can, instead of doing 4 measurements, we can do 3 measurements.
Any further questions? Please do not call my guest a polarized person.
Thank you for the nice talk. Optical methods are usually limited to the surface or near surface. Is there any alternative technique that can reach deeper into the tissue, like microwaves or acoustic techniques that can give you information about similar information like polarization, but about deeper inside the tissue?
Optoacoustics goes deeper. Optoacoustics, photoacoustics, yeah, it can reach, let's say, centimeter resolution. But I can tell you that when we just start our collaboration, we told medical doctors that what we have at hands, it's a seconds because we didn't have this optimized instrument, updated design. It's a few seconds for measurements. The technique is 2-dimensional, yeah, the penetration depth. I can tell you my estimation, of course, it depends on tissue. Yeah? If you measure cornea, which is transparent, you can go deeper. Brain, white matter, from the name of the matter, it's very scattering. So my estimation that we are probing maybe top 100 micrometers. Yeah? And this is so I explained that it's quite slow and it's superficial. And the answer was, we go for it because we don't have anything. We are happy to have this. But when we advanced in the project, when they heard that, oh, we can make it fast. Yes, we need a video rate. Please make it faster. Now they say, because they are removing and they want to see not only lateral borders, but to have an idea when they are approaching, let's say, the end of tumor, when they should stop. We are working on it, but I would say in a visible, we are limited to this like 0.1 millimeter. Photoacoustic probably would do this. A OCT, this in front, yeah, but their image millimeter by millimeter, and they do not have luxury to scan. It's too slow for them. I know that they have a size microscope, confocal microscope, and they even published the results because with confocal microscopy at cellular level, you can immediately see tumor or no tumor. But they told that it's of no use because they cannot scan where to cut. They need to see the border. So wide field, it was also one of the fast, compact, wide field. And yeah, of course, 2D is not best they want, but that's what we can suggest them. And for example, microwave, I'm not sure that you can easily use it in operating room. The white light, it's okay, yeah, to shine because there is light in the surgical environment, but microwave probably safety regulations are different.
Thanks.
Okay, let's thank our speaker.